Hi, I'm Farrukh. I've learned to carry out PCR in my molecular biology module. Today I'm going to carry out an RT-PCR reaction to test for SARS-CoV-2, the virus that causes COVID. Real-time RT-PCR is a powerful technique for detecting specific DNA sequences. SARS-CoV-2 is an RNA virus, so first we have to convert the RNA into DNA with reverse transcriptase. Then we use specific primers that hybridize with the DNA in the presence of Taq polymerase. They copy the viral sequences. By varying the temperature, we can repeat this process, where at each cycle, the sequence of interest is doubled. These are 4 swab samples from the patients. They're in a liquid that has an inactivated virus, but preserved both the RNA and the proteins. I'm going to remove the swab and collect an aliquot into these microcentrifuge tubes. To carry out the RNA extraction, I'm going to use these small columns that contain a matrix that specifically bind to RNA. When I spin the columns in the centrifuge, the RNA will remain on the column. I can discard the liquid that contains proteins and other molecules that I don't need. I now add buffer to the column to wash out any remaining protein, and centrifuge again. Finally, I add pure water that will make the RNA come out of the column and into the collecting tube. I now have purified RNA from the samples. Here I am preparing what we call the master mix. Basically, I'm combining together all elements that will be needed to add to each of the samples. It includes the enzymes reverse transcriptase, Taq polymerase, and the specific SARS-CoV-2 primers, the nucleotides, magnesium, and a buffer. It's now time to prepare the PCR reaction. I first load the 4 samples. I'm also going to load a positive control, which contains SARS-CoV-2 RNA, and a negative control, which has no RNA. I'm now adding the master mix to all the samples that I prepared before. I can now load the plate into the thermocycler. I'll program it to go through 40 cycles of temperature variation. As I explained before, at each cycle, the specific sequences from the virus RNA will be doubled. The machine can detect the doubling of the nucleic acid in real time, and the results are displayed graphically. We can see the curve of the positive control is very similar to that from patient 3. However, patients 1, 2, and 4 are different, and therefore we can say that only patient 3 is positive for COVID-19.